Then MSc Part 2, Semester 4, Unit Paper 1, Unit 2, supramolecular chemistry. Okay, so the next point is the Rotaxanes. The question is, what are the Rotaxanes? Discuss their structure. Discuss their structure. Okay, कसा क्वेश्चन असू शकतो रोटॅक्झॅन वरती, व्हॉट इज द रोटॅक्झॅन अँड डिस्कस देअर स्ट्रक्चर. किंवा एक्सप्लेन देअर स्ट्रक्चर. Okay, the answer is there is the very little free space in center of a cycloalkane unless the ring is quite large. The calculation indicate that the cyclooctadecane is the smallest ring through the which is the CH2 n times CH2 chain can be threaded. The molecule have been synthesized that have the large ring threaded on the chain, the first, and that have the large ring which are the interlock like links in a chain 2, and the former molecule are called as rotaxanes and the later molecule are the catenon. Okay, so the firstly is a rotaxanes, then catenon, and 3 is also catenon. The catenon, Latin catena meaning the chain, are the molecule containing the 2 or more intertwined rings. Is a similarly rotaxanes is a Latin rota meaning is a wheel or the axis, are a molecule in which the linear molecule is the treated through the cyclic one and prevented from the slipping out by the large and bulky end group. And these two classes of a compound contain 2 or more portions that are not bonded to the each other by any valence force, that most remain linked. Okay, these are the rotaxane and their explains their structural features. Okay, the next question is a molecular recognition or catalyst. What is the question? They explain the strategy employed for the antibody catalysis in terms of a molecular recognition. Firstly, the no discussion on the molecular recognition and catalysis can be complete without referring to antibody catalysis. The antibodies are large portion molecule consisting of the 4 polypeptide chain produced by the specific lymphocyte or immunocyte in response to a foreign micromolecules known as antigen introduced in the body. The antigen is the antibodies are highly complementary to each other and form the strong complexes. The small molecule is a heptane when coupled to the carrier micromolecule. Example, a portion become the immunogenic and generate the antibodies with a complementary binding site which act as a highly specific and selective receptor. And these strategies employed for the antibodies is a catalysis are follow for the using the heptanes, which is a resemble the rate determining transition state of the intended reaction, and the binding site of an antibody are to so designed that they selectivity stabilities the transition state through the molecular recognition and thus the bring about the activation energy. For example, the antibodies raised against the tetrahedral and negatively charged phosphate and phosphonate, which are the transition state is analogous of a carbonate and ester the hydrolysis. Then respectively the catalyze this reaction with the accelerated rate. In the second strategy, the antibodies binding site are the so tolerated by the specific haptens that are the reacting the partners are brought into the close proximity as liquid is a highly ordered transition state. In a third variation, specific catalytic groups, example of general acid-base type of may be introduced into the binding site. Thus, in situ elimination of a beta fluoroketone heptanone as a analogous structure, but with the alpha H is replaced by ammonium group. And the yet another variation is antibodies may be raised in the multi-substrate analogous to that. The binding site are can simultaneously bind the cofactor and the substrate to the catalysis, the reaction in which the enzyme fraction. There is no fundamental difference between the enzyme catalysis and antibodies catalysis, both the showing high selectivity, high reaction rate, and saturation inhibition. Thank you.